we have this question, guys. They're saying figure 2.1 shows a section through a human tooth. All right, and the parts are well labeled there. And they're saying question A1, state the names of parts labeled F and G. So F is the enamel. G, where's G? All right, G is the dentine. Dentine. Okay, G is the dentine. Then they're saying number 2, under question A, explain the functions of the parts labeled H and I. The functions of labeled H and what? And I. Okay. So the functions of labeled H. Look at where H is pointing. That area contains nerve cells and also blood vessels. Okay. It contains nerve cells and blood vessels. So you can say nerve cells make the tooth sensitive. Make the tooth what? Sensitive. Okay. And blood vessels in structure H supply the tooth with the food, okay, and oxygen. Okay, now listen, these nerve cells are not only sensitive, are sensitive to what? They're sensitive to pH, they're sensitive to temperature, and also sensitive to pressure. Okay, let's go to the function of the structure labeled I. The structure labeled I. So the structure labeled I, this is a bone-like tissue. It's a bone-like tissue which anchors the tooth in the jawbone. Which anchors the what? The tooth in the jawbone. This function is to anchor the tooth in the jawbone. Okay. Next question. Question B1. This is question B1. It's saying, explain, how many marks? 2, 3. Explain how tooth decay is brought about. Wow. Let me give you full information how tooth decay is brought about. So, tooth decay is brought about when sweet food particles stick in between the teeth for so long and form a plaque. Now bacteria start to feed on the what? On the plaque and respire anaerobically, producing an organic what? Acid. This acid dissolves. To dissolve is to corrode. Dissolves the enamel, which is the hardest part of the tooth, the tough part of the tooth. So dissolves the enamel of the teeth. Okay, forming a cavity. All right, forming a cavity in the teeth. When this cavity reaches the dentine, the tooth starts getting painful. and the pain increases further when the cavity reaches what? The pulp cavity. This is called what? This is called tooth decay. All right, this is called what? Tooth decay. Next question, which is 2. Suggest 2 ways of preventing tooth decay. Suggest 2 ways of preventing what? Tooth decay. You can say avoid eating sugary foods. Number 1, avoid eating sugary foods. Number 2, you can say brush by brushing teeth, okay, with the fluoride toothpaste. With fluoride what? Toothpaste every after meal. Okay, every after meal. That's what was this question. Let's go to the next. Guys, we have this question, and we are told that figure 1.1 shows a plant cell that has been put in a concentrated salt solution. Yes, because this is a result when a plant cell is put in concentrated solution. It loses the cell wall remains in its shape, but the cell membrane loses water. So it loses water. Now A, identify the parts labeled A and B. So this is question A. So part labeled A is cell membrane. Okay, it has shifted because it has lost what? Water. So that one is a cell membrane. B, where is B? B is pointing to those white circles. Those are chloroplasts. Okay, then B1, question B1, they're saying, what happens for the cell to reach this state? It was placed in a concentrated solution. Okay, you can say the cell was placed in concentrated solution. So in brackets you can write is hypertonic solution. Hypertonic solution. Okay, hence it loses it lost water. Okay, it lost water by the process called the osmosis. By osmosis. Question B2. Question B2 is saying, state the term given to the cell in this condition, plasmolysis. Okay, plasmolysis. All right, plasmolysis. Next question is question, what question is this? Question 3, B3. So they are saying, how can this condition be reversed? How can this condition be reversed? So this condition can be reversed by putting the cell in dilute solution. Okay, dilute solution. This dilute solution is usually hypotonic solution. hypotonic solution, such as pure water. All right, next question, which is question D. A, B, C, not D. Question C. They're saying, give one reason why the structure labeled C remained in its natural state. So you can say structure C is made up of cellulose, which is rigid or which is tough. Okay, which is tough. D. Question D. Give two differences between a plant cell and an animal cell. Okay, so you can say now the difference number one, a plant cell has a cell wall, while an animal cell doesn't have. Okay, doesn't have a cell wall. Another difference is that the second difference is that a plant cell, or you can say most plant cells, have chloroplasts. Okay, why would say most? Because some don't have chloroplasts, like root hair cells. They don't have chloroplasts in the roots, and chloroplast is only used for photosynthesis because it contains chlorophyll, which traps light energy. So photosynthesis takes place in the leaves and not in the roots. So root hair cells, they're plant cells, but they don't have chloroplast. Okay, so you can say most plant cells have chloroplast, while animal cells don't have. Okay, don't have. All right, thank you so much. That's what with this question. Let's go to the next question. Okay guys, so now let's look at the human digestive system. Look at a question on the human digestive system. Okay, this is the question, the diagram, and the questions. All right, let's now answer the questions. Now, when you look at the diagram, let's study it first, okay, before we start answering. We have the esophagus, the stomach there, and we also have structure V, W, and X. When you look at V, that is the pancreas. When you look at W, that is ileum or small intestines. X, that is the anus, where ejection takes place. Now let's look at the question. So question A, you're saying from figure 2.1, select the letter where ejection occurs. So ejection occurs at X. Okay, at X. Number 2, select the letter where pancreatic juice is formed. So pancreatic juice is formed in the pancreas, and the pancreas is V. Okay. 3, number 3, you're saying where villi are present. Villi are present in the ileum or small intestines, and W points at small intestines. Okay, let's go to question B. So question B of this paper saying The stomach produces hydrochloric acid. Yes, this we know. State two functions of this acid in the stomach. Okay. So number one, so you can say it kills some bacteria. Okay. It kills some bacteria that comes along with the bolus. Okay. Then number two, you can say it activates the enzymes and pepsin and renin. Activates the enzymes pepsin and renin, and also sets the acidic pH, okay, which is optimum. Remember, the word optimum means suitable. So optimum pH is the suitable pH at which an enzyme works best. Optimum temperature is the temperature at which an enzyme works best. Let's go to the next question, which is question C. Okay, so question C is saying, describe the role of the liver in 1. Digestion. So in what? In digestion. So the role of the liver in digestion is that it produces bile. It produces bile, which contains bile salts. Okay, which contains bile salts. And they can say bile salts help in emulsification of fats. Okay. Then number 2, assimilation. The role of the liver in assimilation. All right. So the role of the liver in assimilation, you can say the liver converts excess glucose to glycogen. Okay, to glycogen. This glycogen is assimilated, so you can say which is assimilated by the cells in the liver. Okay, by the liver cells for storage. Okay, for storage. So now this glycogen is stored in the muscles and also in the liver. The larger quantity is stored in the muscles, then the other one in the liver. Okay, that's the role of the liver, and they usually ask such questions. The role of the liver in digestion: it produces bile, which contains bile, which contains bile. Then bile salts help in assimilation of fats. Okay, then assimilation, not assimilation of fat, emulsification of fats. And emulsification, this is the breaking down of large drops of fats into smaller droplets, all right, by an enzyme which is called lipase. Okay, and fats, when they're converted, when they're broken down, the digestive end products are glycerol and fatty acids. Let's go to the next question. All right, so that's all to this question under digestion. Thank you so much. Hello guys, how are you? Let's revise biology 2009 paper 2 phloem cell. Okay, so we'll answer a number of questions in this series. So let's get started. The question reads, figure 1.1 shows some cells taken from a vascular tissue of a plant. Okay, question A, they are asking to name cells X and Y. Okay, cells X and Y and the structure labeled Z. So the answers, this is question 1A. Question 1A. So cell X, remember they're just saying X cell, not the part. So cell X is companion cell. Companion cell. Okay, is a companion cell. Then cell Y, which is the host cell, this is the phloem. Okay, phloem what? Phloem cell. All right. Then the next question reads, which is question B. What are the functions of the cells labeled X and Y? Okay, so this is question B. What are the functions of cells labeled X and Y? So let's start with the function of X, then the function of Y. So the functions of a companion cell or companion cells, you can say they provide energy, provide or supply energy to sieve tubes. Okay, to sieve tubes. Okay, so cell X provides energy to sieve what? Sieve tube. So because companion cells are living cells. All right, question Y, the function for cell Y, which is the phloem. The phloem transports food to different parts of the plant. So this one can say phloem transports food to different parts of the plant. Okay, of the plant. All right, next question. The question reads, okay, which is 3. So this one is 2. So 3, you're saying, what are the functions, what structure in the human body performs a similar function as structure Y? All right, what structure in the human body performs a similar function as structure Y? As the phloem, meaning transporting food. All right, transporting food to all parts of the body. Which structure does this? So the answer is blood vessels. Blood vessels. Since the phloem transports food to different parts of the plant, blood vessels also transports food substances to different parts of the body using blood. Okay, let's go to the next question. The question reads, which is question C. what two conditions are necessary for osmosis to take place? What two conditions are necessary for osmosis to take place? Now, for osmosis to take place, number one, is that there must be two regions separated by a selectively permeable membrane. Okay, selective permeable membrane. Number two is that there must be difference in concentration. There must be difference. There must be difference. There must be, must be a difference in the concentration. Okay, between two solutions. Okay, between two solutions. So these are the two conditions. Okay, there must be separated by a selectively permeable membrane. Number two, there must be a difference in concentration between two solutions. Okay, next question. Oh, that's all to this question. So 8 marks in total. Okay. Oh, sorry, there is another question that I skipped. The question was cell X, cell Y, and structure Z. So you can say structure Z is sieve plate. Okay, structure Z is sieve plate. All right, so that's all to this question. Guys, we are presented with this question and we have figure 5.1 and 5.2. So they're saying figure 5.1 and 5.2 show the cross section through two different portions of the alimentary canal. Question A, 1. So they're saying A1, they're saying from which part of the alimentary canal were these cross sections taken? So the first one is figure 5.1. 5.1. What's the answer to this question? So for figure 5.1, look at 5.1. In the inner lining of 5.1, you have those protruding structures, those elongations. Those are called villi, and villi are only present in the ileum or small intestines. Okay, so you can say small intestines, all right, which are villi. And figure 5.2, figure 5.2, it has that structure which is the pancreas and the duct. So how we can know that this one is figure 5.2, the section is the duodenum, is because of the pancreas. So it is the duodenum. Then number 2, I'm saying give reasons for your answers in A1 above. We give reasons in A1 above. Okay, so reasons are very simple to this question. You can say reason one, so you can say figure 5.1, okay, is found in the small intestines because of the presence of villi. Okay, those structures in the inner lining. Then reason two, figure 5.2 is found in the duodenum because of the presence of the pancreas. Okay, the pancreas and the duct C. All right, duct C. Let's go to the next question, which is question B. Look at question B very carefully. They're saying, explain the role played by B in the alimentary canal. Look at B, those inner lining. So the role of B, so you can say the inner lining of the alimentary canal, okay, is made up of longitudinal and circular muscles. Longitudinal and circular muscles which contract and relax, okay, thereby pushing the what? Pushing the food. Okay, so in the esophagus is called peristalsis. Okay, pushing the food. Okay, it leads to the movement of the food. C, name two juices passing through tube C. Okay, so look at tube C, look at where tube C is and the other tube which is joining to tube C. The other tube which is joining to tube C comes from the liver. So you can say number one, you can say pancreatic juice, which is the juice produced by the pancreas. And then number two, you can say bile. That's it to question C. Oh, the question has ended. So that's all to this question and alimentary canal. Thank you so much, guys. Go through, write down the solutions, okay, revise. Okay, so guys, let's look at respiration in insects, okay, by answering this question here. And the question reads, Figure 3.1 shows part of the respiratory system of an insect. Okay, and the diagram is very clear there. There is the structure E, carbon dioxide out, oxygen in, and part D there. The first question, which is question A, they are asked to identify the parts labeled D and E. The parts labeled D and E. So, the answers are, so part D is C, the spiracle. Okay, the spiracle. And then part E is a muscle cells. Okay, these are muscle cells. Okay, another question is saying which is question B. Explain how oxygen from the atmosphere is able to reach the tissue labeled E, which are muscle cells. Okay, so oxygen, you can say oxygen enters through the spiracles, enters through the spiracles, okay, which connect to the trachea, okay, which is connected to the trachea. Oxygen enters through the spiracles, which is connected to the trachea, and the trachea, this trachea branches, okay, trachea branches into smaller tubes, okay, many tubes called tracheal, okay. Now these tracheals, they eventually reach, so the tracheal, which eventually reaches the tissue cells or tissues. All right. Okay, question C. Explain how the energy released from respiration in an insect is used. Explain how the energy released from respiration in an insect is used. Okay, so now the energy is the energy released from or in an insect is used for, okay, is used for movement of the abdomen, and wings during the process of respiration. Okay, during the process of what? Of respiration. Okay, it is also used for flying and ground movements. Flying and ground what? Ground movements. movements. Okay, ground movements. And also eating. Energy is also used for eating. Last question, but not the least, which is question D. And the respiratory system of an insect. They're saying, state two ways in which the respiratory system shown differs from that of a fish. Now, this is simple. Two ways. So, number one, you can say in insects, air enters through the spiracle. Okay, through the spiracles in insects. While in fish, okay, while in fish, it enters through gills. Simple as this, guys. So air enters through the spiracle in insects. Then in fish, it enters through the gills. All right. Number two. Secondly, say in insects, in insects, gaseous exchange in insects, gaseous exchange occurs across across the walls of the tracheas. Okay, while in fish, gaseous exchange occurs across across the surface of the gills, across the surface of the gills. Okay, across the surface of the gills. That's all to this question, guys. So go through the solutions. Okay, if it's possible, write down. Okay, so we are given specialized cells. Okay, specialized cells A, B, C, and D. So we are asked to identify A1. So in the table, identify using the letters of the cells, identify animal cells and name them. Okay, so there is one identity here, and this side the name of the cell. Okay, so number one, so we have B. Is, we are starting to use letters, is B. Then what's the name of cell B? Is a red blood cell. Okay, a red blood cell. Then the other animal cell here is, so this is 1, 2. The other animal cell here is C. The name of animal cell C is a motor neuron. A motor a motor neuron. Okay, that's the answer to the first question A1. Then question A2, they're saying, for each named animal cell in A1, state one of its characteristic features. So, feature in animal cell 1. Okay, animal cell 1, the first one, which is a red blood cell, the feature is that it has a biconcave shape. Okay, shape or disc shape. Okay, then animal cell 2, it has dendrons. Okay, dendrons. All right, and as well as the mammalian sheath. Okay, mammalian sheath. All right, the other, let's go to the next question. This is question B. Question B, for cells A and D in figure 1.1, name the substance found in the cell which enables it to perform its specialized function. So when you look at cell A is a phloem. Cell A is a phloem. Then when you look at cell B, cell D is a palisade cell. Okay, it's a palisade cell. So you can say a phloem has sieve plates. Okay, sieve plates. Then the other side, the function. So you have a substance, sieve plates. The function here, sieve plates allow food. Sieve plates allow food to pass through from cells to other parts. Okay, parts of the plant. You can also say another one is a companion cell. Its function is to supply energy to the sieve plates. Okay, so this is A. B, you have a palisade cell. A palisade cell, what makes it to perform its function? There are a lot of parts. You can say a cell wall, you can say chloroplast. Okay, now let's just use chloroplast. Chloroplast. Okay, the function of chloroplast is to trap light energy. The chloroplast contain chlorophyll, which traps light energy used for photosynthesis. Okay, used for photosynthesis. Okay, next question, which is question C. So they're saying we are given a diagram here, an experiment. So they're saying figure 1.2 shows an experiment on osmosis. Okay, so there is solution E and solution F. Okay, solution E and solution F. We have, so for osmosis to take place, like I mentioned in the previous video, where they asked two conditions for osmosis. Number one is that you need to have two solutions with different concentration. Number two is that the two solutions must be separated by a selectively permeable membrane. Okay, so when you look at solution E, let's look at the key first. On the key there, we have the small dot which represent water molecule. A large black dot represent sugar molecule. Then, in which side between solution E and F are more small black dots or water molecules? Automatically, as you can see, solution E has a lot of water molecules. And then the side which has a lot of sugar molecules is solution F. So meaning solution E is diluted. Okay, it has less concentration than solution F. But in terms of water potential, solution E has higher water potential than solution F. Okay, let's now answer the questions. From which solution in figure 1.2 will there be a flow of water molecules by osmosis? So the answer is very simple to this one, which is C1, is solution E, because that's where you have more water molecules, a lot of water molecules, solution E. Okay, next question, they're saying, when will the flow of water molecules across the membrane stop by osmosis? Okay, so this is number 2. So you say the flow of water molecules, stops when a dynamic equilibrium is reached. Okay, the flow of water molecules stop when a dynamic equilibrium is reached. A dynamic equilibrium, what it means, a dynamic equilibrium, when you have equal concentration, when the concentration is equal on both sides. Okay, when you have equal concentration. Okay, concentration, that's when the flow of water molecules will stop. Okay, next question. The question is, why suggest one reason why only water molecules can move across this membrane. The answer is simple, guys. All right, this is question 3, if I'm not mistaken. Yes, this is question 3. Okay, question 3. So they're saying why only water molecules will move. So you can say because water molecules are small molecules. Okay, are small molecules which can be allowed to pass through. Okay, which can be allowed to pass through the membrane but larger molecules and cannot or are not allowed to pass through. All right, because it is selectively, it selects what passes through, substances that pass through, and the other, those that cannot pass through the membrane. All right, next question. That's it to this question, I think. Yes, that was the last question. All right, to this question. Let's go to the next question. Let's look at saprophytic nutrition, guys. By answering this question, the question reads, figure 4.1 shows the growth of muca on shima. Okay. Then the first question, which is question A1, is saying, identify the structure labeled J in figure 4.1. So, structure labeled J is sporangium. Okay, like this. All right. Then, question 2 is saying, explain how structure K obtain nutrients from shima. 4 marks. Okay. So listen, guys. Structure K, so we can say the muca releases enzymes from its hyphae into shima. All right. These enzymes hydrolyze the shima into glucose as the end product. So glucose is the end product. And the structure K, which is the stolon, absorbs this end product. Okay. Then B1, we are saying, the shima was flooded with iodine solution. State the color change that would occur. 1 at L. Okay, at L. Then number 2 at K. The color change. All right. So look at where L is. At L, the color of iodine solution. So we can say the color of iodine. Yeah. The color of iodine solution changes to blue black. Okay, blue black. Then at K, not at K, at M. I'm sorry. At M, so the first one is at L, then the second one is at M. Okay, at L, the color of iodine solution changes blue black, and at M, there is no color change. Okay, there's no color change. So the color of iodine solution just remains the way it is, as yellowish brown. Okay, or just brown. Then C, they're saying, give reasons for the color change observed in B above. So you can say at L, okay, at L, starch was present. Okay. And then at M, starch was what? Was absent. That's it. We have a life cycle of a housefly. So the question reads, figure 3.1 shows the life cycle of a housefly. Okay. Then question A1, they're saying identify the stages labeled G and H. Labeled G and H. Okay, the stages labeled G and H. So when you look at G, those are eggs. Okay, and we want to look at H. You have the first one is a larva stage, the second one is also a larva stage. So you can say H is the second larva stage. Second larva stage. Okay, second larva stage. All right. Then question 2, A2 is saying, explain the changes taking place during stage 1. Explain the changes taking place during what? Stage 1. So you can say not stage 1, sorry, I. Okay, so you can say it increases greatly. Okay, it increases greatly in size. As we can see, it increases greatly in size. Okay, then the other change is that it is reducing. It is reducing in the production of predatory maggots. Okay, in the predatory maggots that feed on other maggots that feed on other what? On other maggots. All right. Another important information that you need to know is that it also reduces. So it is reducing. It is reducing in feeding on the rotting material. Okay, rotting material by sucking the nutrients. Okay, by sucking the nutrients, and also it starts moving using its pads. Okay, these pads are found on the lower side of their bodies. So the changes that take place during stage I is that it increases greatly in size. It is reducing on the production of predatory maggots that feed on other maggots. It is reducing in the feeding of rotting material by sucking their nutrients and moving using pads and found on the lower side of their bodies. Next question. Question is B1. Which stage in figure 3.1 would be most effective for eradicating housefly? Which stage in figure 3.1 would be most effective for eradicating houseflies? So it is the pupa stage. Why the pupa stage? Because it's not moving, so you can just spray insecticides. Okay, at this stage it is not moving. All right, question B2. Question B2 is saying, using a named example, describe the role of housefly in disease transmission. Okay, named example meaning example of a disease. So you can just say houseflies are vectors. are vectors for pathogens, that cause disease, they cause cholera, dysentery, and also typhoid. Okay, that's all to this question. The life cycle of a housefly according to this question. Thank you. Okay guys, let's look at specialized cells in plants by answering this question. Question 1, so they're saying figure 1.1 and 1.2 show the structure of specialized cells in plants. Figure 1.1, when you look at figure 1.1, it is closed, the guard cells are closed. Then figure 1.2, the guard cells are open. Let's go to the question, which is question 1a. They're saying identify the parts labeled, the cells labeled A and B. Cells labeled A and B. Okay? So A, look at where they're pointing. Those are epidermal cells. You can say epidermal cells. Then B is pointing to the guard cells. So B are guard what? Guard cells. Also B is a guard cell. Okay. Now, identify the parts labeled C and D. So this is A1, then A2, the parts labeled C and what? D. So C are those black dots, and these are plant cells. So C are chloroplasts. Chloroplast. Okay. Then D, where is D? D is just the space in between those, that is the cytoplasm. The cytoplasm. Then let's go to B, question B1. They're saying, which figure shows a closed stoma? Figure 1.1 is disclosed. So figure 1.1. Second question. Explain. Wow, I like explaining. I don't like one-word answers. I like explaining because it requires someone to think. Okay? And this is also called deep learning. Explain how the opening of a stoma is brought about. Wow, nice. Okay? So you can say, during, so I've just abbreviated K here. So we can say during the day, potassium concentration, okay? Potassium concentration, this is the short form of concentration. I've just written K for potassium. So during the day, potassium concentration in the vacuum of guard cell increases, okay? Increases. And this lowers the water content, okay? Lowers the water contents in the cell sap, which leads to the flow of water from the epidermal cells to the guard cells. This inflow of water raises the target pressure inside the guard cells. This target pressure causes, the guard cells, this is supposed to be guard, all right, the guard cells to curve in such a way that the stoma opens. Okay, the stoma open. Okay, stomata is singular, stoma is plural. So this is how the opening of the stomata is, or the stoma is brought about. That's do you have any other question here? No, that was the last question. That's it. Thank you so much. So guys, we are given this question, and the question reads, Figure 2.0 shows cells taken from a vascular tissue in plant. Question A1, they're saying, identify the specialized cell, figure 2.1. That we've already answered this one previously. This is a phloem cell. Okay. Then 2, they're saying, name one feature in 2.0 which enables you to identify the specialized cell. 1. Name one feature. So the feature is the companion cell, which is letter B. Okay. Which is a companion cell. Okay. Which is a companion what? Companion cell. The other question. Which letter in figure 2.0 correspond to the following structures? So 1, 2, 3 correspond to the following structures. 1, nucleus. Which is the nucleus? So nucleus is A. Okay. Then number 2, cytoplasm. Cytoplasm. So cytoplasm there is D. Okay. D is cytoplasm. Then let's go to question B. Question B. Which letter? So give two functions of the cell labeled B, and the cell labeled B is the companion cell. So you can say, number 1, a companion cell provides energy to the cell. Okay. Number 2, apart from providing energy, okay, it also supply the cell, okay, with the enzymes. The role or the functions of a companion cell. And then the last, is it the last question, or we still have other questions? Question C. With reference to the specialized cell in 2.0, C1, name the process by which substances are transported in the specialized cell. The process by which substances are transported in the phloem is called translocation. Translocation. All right. Then number 2, name 2 substances transported by the specialized cell. 1, there is sucrose. 2, there is amino acids. Okay. Amino acids. Next question. Oh, that's all to this question. These are the solutions. Thank you so much. Let's answer the first question. First question here in GCE 2017, part 2. So we are given this table. We are asked to complete the table, copy and complete the table. Complete the table below showing arts of microscope and their functions. So part 1 here, we're saying which part collect and reflect lights onto specimen on the stage. This is the mirror. Okay, the mirror. Then 2, okay, this is the first question, question 1A. Then 2, they're saying the diaphragm. So meaning, what is the function of the diaphragm? So the function of the diaphragm is to control the amount amount of light, not only just amount and brightness of the light, okay, of the light falling into the specimen, okay, falling into a specimen. Number 3, which part of the microscope magnifies the image further? This is the eyepiece. Number 4, the stage. What is the function of the stage? So the stage, this is where the specimen is placed. So we can say this is where microscope slides with the specimen are placed. Oh, we still have another one, the 5th one. Which part of the microscope brings the image into sharp focus, especially when the specimen is being viewed at high magnification? This one is a fine adjustment knob. Okay, the fine adjustment knob. Then there is question B, and question B is saying state 3 parts of the plant cell which can be easily seen under a light microscope, not an electron microscope, but a light microscope. Okay, cell wall, of course. Number 1. Number 2, cell membrane. Number 3, the nucleus. Okay, the nucleus. 2016 paper 2 question 1 is under cells. Okay, so the question reads, figure 1.1 shows an animal cell as seen under an electron microscope. Okay, so everything is detailed and everything is labeled there. Is seen. The first question is saying, identify the labeled parts F and G. If you look at F, those are endoplasmic reticulums, but because it has ribosomes, it will be rough endoplasmic reticulum. Okay, that is F. Then G is the nuclear membrane. Okay, nuclear membrane. Then B, question B, is saying, state the functions of part labeled H and I. The functions of parts labeled H and I. So you can say H this is the site where respiration takes place. Respiration takes what? Takes place. Okay, these sausage like structures. Then number I, the function of the parts labeled I is the cell membrane. So you can say, since it is selectively permeable, so you can say it allows some substances to pass through it as it is selectively permeable. Okay, as it is selectively what? Permeable. The other question, do you have other questions? Question C, yes, we have. Suggest two cell parts in which would be present in figure 1.1 if it was a plant cell. So if this was a plant cell, two parts that would have been present. The first one is a cell wall. The second one is a large vacuum. Okay, so that's it to this question. Let's go to other questions. So second question, which is question 2 of 2016, is saying figure 2.1 shows the effect of pH on the rate of enzyme characterized reaction K and M. All right, we have K and reaction K and M. So the question reads, the first one, A, what term is given to pH at J and L? So that is a peak at, so that is that's where it's working best. So it's called optimum. Since this is pH, so we're going to say optimum pH. Okay, optimum. All right, pH. Explain why the rate of reaction K and M goes down after J, after point J and L is what? Respectively. Okay, respectively. So there you can say this is B. So reaction K decreases because the enzyme stops working above 2.5, above pH of 2.5 as seen in the diagram there, 2.5. Okay, then you can also say reaction M decreases because the enzyme decreases because the enzyme stops working or gets denatured, stops working at the pH above, that is 11, above 11. Okay, above 11. Then question C is saying name the region on the alimentary canal where K, reactions K and M takes place. So K, if you look at K, that those are in acidic condition. Okay, acidic condition, and then M is that is alkaline. So K, reaction K in the alimentary canal, this would be in the stomach because that's where it's acidic. Then reaction M You can say in the mouth or duodenum. Okay, duodenum or in the mouth. So you can just write one of these. Then D is saying state one food nutrient which can be catalyzed by reaction in reaction K. Food nutrient, those are proteins. Okay, proteins, because proteins are first digested in the stomach and the stomach is acidic. So D2 is saying, oh, this is not the first question. D1 is saying, suggest one enzyme which can catalyze reaction M. Reaction M there in, so you can say maltose or amylase, because amylase works best in alkaline condition, usually in the mouth. Then number 2, saying state one food nutrient which can be catalyzed in reaction K. In reaction K are, you can say, proteins. Then E, state two factors other than pH which affect enzyme activity. You can say 1, temperature, 2, inhibitors. Okay. Also enzyme concentration. You can just write 1. Enzyme what? Enzyme concentration. Okay. So that's all to this question. Let's go to the next question.